So, hello, I'm Mridul Mohan from group number 324. I'm here today with two topics. So, the first topic is mechanochemical processes in thermodynamic. So, let us start. Mechanochemical processes are those where mechanical work like muscle contraction and molecular motors is coupled to chemical reactions. Example in energy release and consumption of something. And thermodynamic studies the energy transformations in these processes, helping to understand how biological systems harness energy for work. In the biophysical significance of these are the muscle contraction, especially the sliding filament theory explains how muscle fibers contract, converting chemical energy ATP hydrolysis into mechanical work. The thermodynamic principles helps us to understand the efficiency of these processes and factors affecting muscle force generation with image of muscle contraction. In molecular motors, the proteins like kinesin and myosin act as a molecular motors, transporting cargo within cells. Thermodynamics helps us to understand how they couple ATP hydrolysis to movement and how the efficiency is influenced by the factors of load and temperature. Enzyme kinetics. So, in the enzyme kinetics, the biochemical reactions often involve thermodynamics helps us to understand how these changes affects the reaction rates and overall efficiency of enzymatic processes. In key concepts, the free energy is the main thing. The energy that is available to work in a system changes in the free energy of Gibbs energy drive mechanochemical processes. Coupling, the main link. This link of energy releasing reactions like ATP hydrolysis to energy requiring processes like muscle contraction. Efficiency, the ratio of useful work output of energy to energy input in a mechanochemical processes. So here are this the we are going to topic 2, the electronic conformational and interactions. So what is it basically? These interactions describe how changes in the electron distribution within a molecule can influence its 3D structure and vice versa. So its biophysical significance include the protein folding. The intricate folding of proteins into their functional shapes is heavily influenced by electronic interactions between amino acids. These interactions stabilize specific conformations, determining the processes of protein function. Enzyme catalysis. The active site of an enzyme often undergoes subtle conformational changes upon substrate binding. These changes are driven by electronic interactions, optimizing the enzyme for catalysis. Signal transduction. Many signaling molecules like neurotransmitters bind to receptors, triggering conformational changes. These changes are often initiated by shifts in the electron distribution, leading to downstream signaling events. And in this, the key concepts of this are the first one, it comes the charge distribution, the main portion of this. The distribution of electrons within a molecule, which can be influenced by factors like bond polarity and resonance structures. Electrostatic interactions, attractive or repulsive forces between charged groups within a molecule affecting its conformation. Hydrogen bonding, a type electrostatic interaction involving a hydrogen atom shared between two electronegative atoms, playing a crucial role in stabilizing biomolecular structures. So, in this, when we are deeper diving into this topic, it's basically the electronic conformational interactions and the fundamental to understanding the behavior of biological molecules, particularly proteins and nucleic acids. They describe the intricate interplay between the electronic structure of a molecule and its three-dimensional conformation. So, in this, the key concepts always include the charge distribution, the arrangement of electrons within a molecule, which can be uneven due to electronegativity and differences between atoms. So, the electrostatic interactions also attract or repulsive forces between the charged groups within a molecule. These interactions include hydrogen bonds, ionic interactions, and van der Waals forces significantly, which involves molecular conformation and stability. So, in conformational flexibility, the molecules, especially biomolecules that are rigid structures, they possess a degree of flexibility, allowing them to adopt different conformations. The ECIs are the stabilized specific conformations, making them more energetically favorable. Electronic excitations and photoisomerization which observation of light can excite electron to higher energy levels, leading to changes in the charge distribution. This can trigger conformational changes as seen in the photoisomerization reactions, which can crucial in the vision and the photosynthesis. So in this, the main it comes as a protein folding. It's a complex process of protein folding which is driven by the delicate balance of forces including ECIs, the electrostatic interaction between amino acid chains and the guide that folding processes, ensuring the protein adopts its native functional conformation. Incorrect folding can lead to protein misfolding diseases such as Alzheimer's and Parkinson diseases. We have heard it lots of time. So next is the enzyme catalysis. The enzyme catalysis reaction by binding to the substrate and stabilizing transition states. ECIs play a crucial role in substrate binding, catalysis, and product release. The active site of an enzyme often undergoes conformational changes upon substrate binding, facilitated by electronic rearrangement. And it's crucial for DNA structure and function. How it's crucial? The double helix structure of a DNA is stabilized by the hydrogen bonds between the complementary base pairs. These hydrogen bonds are influenced by the electronic properties of the bases, ensuring accurate DNA replication and transcription. So in signal transduction, the receptors, which are often proteins, undergo conformational changes upon the ligand binding. These changes induced by the ECIs initiate a cascade of signaling events within the cell. So in experimental and computational techniques, it includes the spectroscopy. The techniques like NMR, IR, and UV-Vis spectroscopy can provide insights into the electronic structure and conformational dynamics of biomolecules. It also comes X-ray crystallography, the technique which reveals high resolution structure of the protein and nucleic acids, allowing for analysis of ECIs. Computational chemistry. The quantum mechanics calculation can be used to study the electronic structure and energy landscapes of the biomolecules, providing valuable information about the ECIs. So, by concluding, I am saying that by understanding the intricate interplay between the electronic and conformational factors, researchers can gain deeper insights into the mechanical underlying biological processes and develop novel therapeutic strategies. So, that's all. Thank you. Thank you so much for listening. Bye bye.